Okay, sorry, I was muted too. Okay, so now that we went over the microscopes, were there any particular questions that you guys might have had either about the microscopes, what we went over today in lab, anything you went over on Thursday in lab, um, anything at all? Are you guys also pretty good on anything concerning the media plates, like the differences between EMB, TSA, and MSA? How are we feeling on those? Feeling good about the everything? Um, the one thing that I did want to go over with you guys one more time is that there were a lot of questions in lab about the streaking for isolation technique, and it's going to be one of those things that you keep with you like the entire time in your class. Um, and you'll see that sometimes your plate doesn't always look perfect or it's not always isolated. Um, and that could be in part because of your technique. And there were a few cases today, like it's your first time, it's okay. I just want to be able to help you guys um, get through it. And if your plates look good, great. Keep doing what you're doing. But if your plate maybe didn't look right, or if Dr. B gave you a critique and you don't know how to interpret it, I can help you with that. And I can show you guys kind of the proper way to do things like that. I know that I've seen some people in class take their dish, right, and the auger side up. They take it, they open it up, they put the lid down on the table, and then the auger on the table. And then they start streaking on the table. And then I've actually seen people put their loop after touching their plate and all of their bacteria back down on the bench. This is why no food's allowed in class. But all of those things are big. Please don't do that. Um, so, you know, I don't know if it was one of you guys. But if you guys do need help, or if you guys resonate with, oh, maybe I made that mistake before, or things like that, it's no shame. Nobody has done this before, and it's very fast paced, and you're expected to pick it up just by looking at it. If you guys need a second, I'm here to help you guys. Okay, if you guys don't say anything, then I'm gonna assume you already know, and then I'm gonna quiz you about it. It's good practice for when it comes down to the test, because
Could you go over it really quick?
Yeah, of course. Okay, so do we need help on like the streaking or like the setup?
Like the setup, like holding the plate and.
Okay, I got you. So let me see if I have like a good enough example. If not, I'll just kind of roll with it. Okay, this is probably a terrible example, but this has a cap and this has a body, right? So I want to show you guys, and I'll kind of angle it down at the table too. I'll kind of show you guys how I guess I would hold things. So let's say this is my inoculation loop, and yeah, let's put this in frame a little bit, and this is my incinerator, right? So I will have my plate here. My inoculation loop can be on the table right now if it just came out of my drawer. But if I'm going to, let's also say that this is my sample, right? I'm gonna put that over here. Say this is my sample over here, right? And I need to pick from this. So I'll put my loop into the incinerator, and then I'll just tap whatever colony I need, right? Now this is going to be hard because this cap does not come off easily. But usually what you want to do is just pick, the auger side up and turn it over. So the cap is staying on the table. So when you take your auger side up, you'll do your one quadrant and put it back down, put the lid back on, back into the incinerator immediately. Whatever you're touching could or could not be hazardous. So we don't want to, put it down on the table and let it sit there, or we don't want to fling it around, especially if it's from the broth. It can aerosolize very easily. So straight back into the incinerator, right? And then we'll continue to do that in order while we're streaking, and we do 1, 2, 3, 4. But always, it's going to be incinerator. Then you're going to pick up, right? You're going to pick it up with the cap still on the table, and you'll streak. And then once you're done, straight back down. Auger side up. Also, when you draw from this, you don't need it anymore. If you really want to, you can incinerate your loop and leave it on your bench and put this away. You won't need this original sample anymore if you're just streaking for isolation. So does that make sense? That is the most sterile, clean way. Of course, I'll also have gloves on, but that's the most clean way to do this. And then always make sure that this cools enough and that you keep it in the incinerator long enough. Does that make sense?
Yes, that makes sense. Thank you.
Okay. And then we're good on streaking too, because it's kind of funny. There was somebody who labeled theirs 1, 2, 3, 4, but instead of going 1, 2, 3, 4, they went 1, 2, 4, 3. They still got isolated colonies. Kudos to them. Their technique was good, but they just didn't go in the right order. So, things like that, it works, but like we need to make sure that we do it the way that Dr. B wants it, because that's how you're going to be tested. And you don't want to say, oh, well, I'm picking an isolated colony and it's from the second quadrant, unless it really is an isolated colony. But when you think of where is my isolated colony going to be, you're going to want to think quadrant 4, because that's ideally the most spread out, right? Like I was trying to think of a really good analogy to put to the spreading of the bacteria, but the best I could do is probably like paint. So if we take a paintbrush and we start somewhere and we keep going back and forth, back and forth, back and forth, eventually the amount of paint that we start at the top, it's going to be a lot more dense, it's going to be a little colorful, things like that. But as we keep going, it's going to get patchier. We can see more of the background, and it's going to become speckless. We're not going to have a whole lot of paint left on the paintbrush, right? So hold on, I have to check. But like, think of that when you're doing your streaking for isolation, except now instead of just having the paintbrush keep going, you're going to paint a little section, wash your brush, and then take a little bit of paint from that section and move it. And as it goes on, you're going to get less and less paint, right? Can we get practice quizzes for the chapters that we go over, or even lab quizzes? Yeah, I can go ahead and make some of those things for you guys that are just sample questions, and it'll be pretty geared towards the either lab or lecture, or somewhere in a mix of those two things, because she might take something that's like, because like a gram stain, it's part theory, part application, right? So she might take that and put it on the actual theory side of the test and the actual lab portion of the test, right? So if we study kind of concepts like that, we'll need to critically think about those. So don't always think that it's going to be so black and white. Sometimes there is a gray area with some of the material you guys learn. So with that being said, I will most likely post those in the remind. And if you guys want to go over them in your own time, or if you guys think, this specific question is really hard and I didn't understand it at all, we can go over it on a Zoom. If your question is probably somebody else's, that's the number one thing. That your question is probably somebody else's. So, don't be afraid to ask anything, because somebody will probably have the same question, and if I answer it, then that's two people I just helped. So, how are we feeling? Is there any other questions about anything that's been going on that we can think of? I think probably starting next week, I'll get more of the differential media questions, but can you explain how we know if an image is gram stained or not? So for the purpose of this class, I don't believe that you will be tested on your ability to look at a picture and tell if it's gram stained, but you will be tested on your ability to say if it's gram positive or gram negative. So basically what she's gonna say is, this is gram stained, is it positive or negative? And that's it. They won't be like, so tell me about this picture. You think it's gram stained? Interesting. It won't be like that, I promise you. What you will have to know is the differences between a gram stain, the Evil Knievel one, the Medieval, I think it's Medieval, yeah, the Medieval one, the shoot. See, even I don't remember them. It's been so long, but you'll need to be able to tell the differences side by side. Now I promise you, it's easy in terms of the different types of stains. When you look at them, you're like, oh yeah, these are all different. But it's remembering the names and what are used is the hard part. Remember that memorization. So that's what your interpretation of a graph will always be, what type of stain was used, and it's only going to be the bank of sample questions that you have, and they all look very different. But we can go over those in detail probably next time. I'm sorry, because it's already 6 and I don't want to hold you guys overtime if you guys have any obligations, but we can go more in depth about kind of those types of things. But all stains are going to look like stains. It's going to look like a bacteria that has pigment on it. So we can go over the different types of stains on Wednesday if you guys are available, or I can stay overtime a little bit. What are we thinking? Does overtime sound enticing? If not, it's okay. My feelings are not hurt. How we feel. Next meeting? Either one. Well, look, let's see. So Jamie, since you asked the question, I can go over it now. And if everybody else wants to leave, they can. But if you want to stay, you also are more than welcome to. I will also go over it on Wednesday. Does that sound like a fair deal? I mean, repetition makes it stick. Okay, I'll go over it now. All right. So let me open up the PowerPoint for you guys, because this is where the best examples are, and you guys have access to these PowerPoints. No problem. All right. Here is the PowerPoint. I don't need this. Let's go full screen. Okay. All right. So let's go over simple staining. So I'm gonna be honest, this was the hardest part of trying to stay awake was this lecture. I actually did fall asleep in class to this specific lecture. I remember because I had to go back and make it all, but that's another story. So with our simple staining, I want you guys to know that if the stain has a name, like that we're gonna look over in the like if it has a name associated with it, so like the medieval ones and the gram stain, those are not simple stains. If they have somewhat a special name to them, they're not simple stains. So simple staining is a single dye, and it's going to be it's either going to have a positively charged cation in the dye or a negatively charged anion in the dye, and that is the portion that it's going to be attached to is called the chromophore. So mouthful. So so the so that's going to be the most like that's going to be the big highlighted part is going to be is it positive or negative? Is it going to be a and you know what? Another big thing that I did was that I changed up cation and anion. So how I remember it is that cats always have a lot of energy. Just think about a cat like on I don't know, what is it called? Catnip. Cat on catnip. If we have a cat on catnip, then they're haywire. They're going all over the place. They are really positively charged. But if we and like their activity is up there, right? But if we have anion, I always think that Anna's really tired. Anna wants to go to bed, like a person, like they're tired. So their energy levels like through the floor, right? They are negatively charged. They might even be grumpy. So cation, super energetic, a lot of positive energy. Anion, mellow, negative, you know. That's how I kind of compare it. And that's what helps me. If you guys have a different association, if you guys want to share it, you guys can. If it's better than that, it probably is because there's not a whole lot. At least I thought I could work with for the cation anion thing, and it still didn't work that well for me. But if it sticks with you, then kudos. Anywho, so chromophore, right. I'm gonna say that you'll at least need to be familiar with the word oxychrome portion, and that is the part that ionizes the chromogen. Chromogen attaches to the fibers and tissues. The only reason why we use simple stains is for the contrast. Remember, bacteria is clear on a light microscope. We can't see anything. So, that's why I was like, I don't know if this is necessarily a gram stain because they could have just used one of the stains used in the gram stain because there's two different ones that we use in the gram stain, right? They could have just used one and that makes all bacteria appear to be, gram positive or gram negative, or they could have used the other one. So it's kind of hard to tell sometimes. And when you look at like when we saw those slides in class of the blood, it's going to look similar where different cells are going to have different colors. It's going to look differently. They're going to have different shapes, sizes, arrangements. So that's what the stain is used for. If we see clear on clear, we can't see anything. We can't tell what it is. So that's why we use stains. So I know simple, basic synonyms, right? They're pretty much one and the same. But in this class, it's complicated. Basic stains are for the positive charge ones, okay? So bacterial staining, the positive charge on the chromophore is attached to the negative charge on the bacterial surface, and it stains the cell wall. So, or not the cell wall, excuse me. It just stains the cell because it could or could not be a cell wall. Because we always love those atypical ones. But this is going to be the oh no, my battery's gonna die on my computer. I have like 10 minutes, so we can get through it. So basic stains are gonna be positive. I try to think of like I don't mean to be like mean, but like a Brittany, you know, like a Brittany. She's like all positive, bubbly. She probably likes to be basic and go to Starbucks and get certain things, and she likes doing this and that. And think about like Mean Girls. Like they're all basic, basic. Like they're all just kind of basic. It's just they're all bubbly people and you know, whatever. So
The gram negative and the gram positive, how they're pink and purple.
mhm.
So I like to think of like the pink one is negative. Like I think of pink like you got your period, so you're negative. That's
Yeah.
that's how easy it took me to remember it.
Yeah, that's a good one. Let me think. For pink, purple, I don't know. I think it was just more. It stuck a little better with me, so it was like, you know, I kind of just said, okay, purple is positive and pink's negative. So, but I still second guess myself. But that is a good one. I don't know if the guys will use it, but the girls know. All right. So basic stains are the positive from on the chromophore, right? Negative stains are or acidic dots. So it has the negative charge chromophore. Um, there's kind of no other way around it. You either have to come up with like a super niche like word association with it, or you could just have to like repeat it to yourself a million times in order to understand or like get it. It's just memorization. Um, but like the negative stain, when you look at it, it's going to leave a negative space where the bacteria is. That's the one thing that you do need to know theory wise. The negative stain leaves a negative space where the bacteria should be. I'm sure you guys like know a little bit about like camera negatives, how it's inverted, right? That's kind of how I would view this one. Because if we look at this one, what if it was inverted? Uh, you know, the bacteria would look like the color. That's what it looks like on this one. The bacteria has the color. This one, bacteria doesn't have the color, right? So we understand the simple dyes, both the basic and the negative slash acidic. Good on these ones? Okay. How are we on morphology? How are we on that, by the way? I don't hear anything, or I don't see any chat. So I'm gonna assume you guys at least know a little bit about it, are comfortable looking at it, and you're not experts, but you're definitely not unknown. So we went over these a little bit. Spirochetes. Number one is syphilis. I will always drive this that STD protection is like number one here because we have syphilis, gonorrhea, herpes, and HIV. All of those are like booming in Bakersfield. So please, no Tender for a little bit, no Bumble, no nothing, because who knows what they have out there. I'm sorry, but before you even think about doing something, you got to get tested because it's just nasty world we live in. So let's see. So this is the Medievals one I was talking about. So Medievals, you're gonna need to know Congo red first, and then the counter stain is gonna be, or sorry, this is switched, so it should be carbofusion first, and the counter stain is Congo red. That's my bad. These in order on the screen, they should be flipped. You'll need to know that, and then you'll need to know that this is meant to picture the capsules. So if you see capsules on a stain, that means that this is most likely medievals, right? Let's see, for the Schaefer-Fulton spore stain, it's kind of in the name. You're going to see these endospores, right? They're just going to look like green or black little dots in the bacteria. It's going to look kind of strange. So you'll need to know first, malachite green, second, safranin. Okay, these are just names to you, but they'll make sense, I promise. The acid fast stain, you'll need to know that this is primarily for mycobacterium. And the first stain is carbofusion. See how we used it twice already? And the counter stain is methylene blue. Okay. You guys will learn about the gram stain more. I don't mean to rush, but my computer is about to die and I don't want to be cut off in the middle of saying something and you guys not get it. But is there any questions with what I just covered? It's mostly just memorization for those ones. This is going to be in your exam 1 module on the right side column. I believe it's lab number 4. Yes, lab number 4, staining. Are there any questions? All good? Okay, one other thing that I did want to kind of drive in really quickly is the how we prepare the specimen for staining. So for this smear is going to be really important. We'll go through the whole like, oh, how do you write your circle? How do you label it? Things like that. We'll go through that. But when you take your slide and you have your circle, it's going to be 1 drop of water, and you're going to get even less than what you would get for a streaking for isolation. You want to get barely anything, and you want to fill in your circle. You want it to look even less cloudy than the one in the middle. The one in the middle is like okay, but you want it to look less cloudy. And so when you heat fix it, it might kind of fade away a little bit, but I promise you it's still there. There is a lot of bacteria that you can grab. And I think this lab will help you guys visualize it. And once this lab passes, there will be no more overgrowth on isolation plates. You will be able to isolate colonies every single time. It kind of helps visualize it a step further. That a little goes a long ways. But I think that's all I have. What do you guys think? How are you guys feeling? Let me see what percent of my computer is at. I have 8%. May or may not get cut off. How are we feeling?